Okay, so nucleic acids. Nucleic acids are basically macromolecules that store information. A macromolecule is basically just a large molecule, macro meaning big. The two basic nucleic acids that you are familiar with is DNA and RNA. DNA stands for deoxyribose nucleic acid, and RNA stands for ribose nucleic acid. Um, and if we look at this first one here, this first image here, this is a picture of RNA. And over here we have our phosphate group, which is basically, it's a phosphate group, so it has your phosphate and your oxygens attached to it. Then in the center here, this pentagon is ribose, and that is a sugar. And then over here you have your nitrogen base, and that can either be one of the following four. It can be adenine, which is abbreviated A, guanine, which is abbreviated G, thymine, which is abbreviated T, and cytosine, which is abbreviated C. As you can see, the adenine and guanine are double rings, which means they have two structures. You can see that one, two. And they are known as purines together. And an easy way to remember that is Ga. And that's sort of what babies say, and babies are pure. And they say Ga, and they always say Ga twice, so there are two rings. If that helps you, then that helps you. If not, then it's sort of stupid. And then you have your pyrimidines, which, I don't know, they're just pyrimidines, and they have single rings here and here. Those are not for this. Don't mind that. And so what you have is you have this, which is DNA. And DNA is the information storage for the body. It gives each living organism, it tells each living organism's proteins how to make that organism's cells and structures and all that sort of thing. And if you see here, we have our phosphate groups running down this end, and same on the other end. And then we have our sugar in the purple, and then we have our bases, our nitrogen bases right here. And as you can see, we have thymine or T, and I'm just going to refer to them by the first letter from now on. You have T always bonding with A, and C always bonding with G. So T always bonds with A, and G always bonds with C. Now, why is this important? Well, this is important because when they split down the middle through right through here, the way that the body knows how to make the other side is because of this right here. T always bonds with A, and C always bonds with G, and G always bonds with C, and vice versa. Now, the other thing that's true with these bonds here is that T and A always have a double bond, and C and G always have a triple bond. And they're held together with hydrogen bonds. And they, again, this is DNA, which is nucleic acid. It's all basically the same thing. And that stores the genetic information for people. Now, what does that do? What does DNA create? What does it make? It makes the, it tells the cells to make proteins. And here are proteins. Proteins are known as polymers of micro acid monomers or something like that. Basically, It's a chain of amino acids. And amino acids are this. These are all your amino acids. And these amino acids are taken from your DNA. And your DNA codes, it's actually your RNA. Your RNA codes these here. These are called codons, and you see that they have similar letters. The U and the T has been replaced, but basically it's the same thing. And those create amino acids, which is your valine and leucine and all these other ones. And then they come together in these chains of the amino acids, and you can see them lined up right along there. Now, an amino acid is basically a molecule that has a carbon at its base, a central carbon it's called, and then a bunch of functional groups on the side. So here, right there, we have an amino acid, and right there we have acetic carboxyl group. And I don't know if you guys are going to need to memorize those. We had to memorize 5 of the most common ones. And those come together, and those form long amino acid chains. Now, an amino acid chain isn't yet a protein. It then has to bond with itself and fold up. And it folds up from a loose, long string like this structure here, and this structure here is the same, and then it goes to this structure here, and it gets all folded up into its final shape. Now this shape has this protein has 2 main shapes. There are known as alpha helix, which this spinny part here is, and then there's this other part called a beta sheet, and that's that flat part there. And those are just names for the type of structure that exists for them. Now those are joined together with polypeptide bonds, and those hold the beta sheets and the alpha helix together and all of that. Now, the problem that we get with proteins, and this causes a lot of different diseases, is first mutations, but that's a whole nother thing, but is something known as denaturing. And denaturing is when, due to heat, pH, or other things, but those are the main 2, the protein unfolds. And since the polypeptide bonds stay intact, it doesn't unfold back into this. It unfolds into some circuitous, sort of messed up, still sort of bonded into parts of it, but not really sort of junk there. And so that denatured protein is now junk, and it could damage the cell, and so that needs to be gotten rid of. And so that's denaturing. Proteins are enzymes. They also are physical structures. And then the main big one is transport proteins. And a transport protein is, let me show you one, is something that transports something between 2 sides of the cell membrane. And I'll send you a video that really demonstrates this very well, because there are a bunch of different types of transport proteins, but this is basically the idea. The outside is here, the inside is here, the cytoplasm, and this is your protein in the middle, and you can see it's all folded up like a protein. And then this is your cell membrane, this purple part here. And so you can see these chloride dots come through and they go to the other side. And normally chloride wouldn't be able to pass through the cell membrane without this transport protein. And there are a bunch of different types of proteins and a bunch of different types that are created. This one is a CFTR, and that has to do with CF, but you don't need to know that. So where does the diversity come from? Again, it comes from the amino acids, which are found here. All of these dots are those. I just found this drawing tool, so it's really cool. And those are proteins. Next, we'll move on to lipids. But all the lipid stuff is down here for some reason. Okay, so this is lipids, but the photos are in the nucleic acid part, but it is lipids. So lipids are a fancy word for fats. So fats include oils, so that stuff that you put on your salad, that stuff that drips off your burger, that's all fat, that's all lipids, it's all the same thing. Lipids by nature are hydrophobic, and that basically means that they are water-fearing. It's why you have to shake up your salad dressing, because they've separated into lipids and waters. You have three main types of lipids. You have your monoglyceride, your diglyceride, and your triglyceride. And your monoglyceride is made up of a glycerol molecule, which is this green part here. I will circle it right there. That is your glycerol. Oh, that didn't work. But that green part's your glycerol. And then each one of these long chains here is a fatty acid chain or a hydrocarbon. Get rid of those don't really work. You can move it. Oh, look at that. And that is your glycerol, and that is your fatty acid chain. And so when you have one, it is a monoglyceride. When you have two, it is a diglyceride. And when you have three, it's a triglyceride. Now, you've probably heard of the term saturated fat and unsaturated fat and where that comes from. That comes from having all of the possible hydrogens that exist. Now, let me say that again. In the fatty acid chains, in the hydrocarbon chains, you have hydrogen and carbon. You have a lot of them. And to be unsaturated, there is fewer than the maximum number of hydrogens at the double bond. And and those fatty acids that are saturated, they contain the maximum number of hydrogen atoms, and it gives it a straight shape. So if we look at this one here, zoom in, this here, right here, is, I want to be correct with this, is unsaturated. And that, because it's bent, you can tell that it's unsaturated. And the one that has all of the carbon, all of the hydrogen possible, are straight right there. Now the bond that connects the fatty acid chains or the hydrocarbons to the glycerol to create the lipid is a dehydration reaction. And that is, we can zoom in again right here, that is here where you have an OH and an H from the glycerol and OH from the fatty acid chain come together and produce water. That's why it's called dehydration. And then that forms a bond between the fatty acid chain and the glycerol. Yeah. Obviously, those diets with a lot of saturated fat can have heart problems. Some saturated fats include butter, cheese, meat, basically anything that's solid at room temperature. And then unsaturated fats are your plant oils and that sort of thing. Now the interesting thing, and they're generally liquid, or if they're oils, then they're unsaturated. Now, if you've heard the word hydrogenated or unhydrogenated, that means to forcibly put in the hydrogen into unsaturated fat. So if you were to I'm sorry, I can't say it. If you were to put the hydrogens in, then it would become saturated. And then when you do hydrogenation, you create trans fats, and those are bad. And trans, if you remember it all from chemistry, is merely saying that the location of the hydrogens are on opposite sides of the molecule. If it was cis, then it would be on the same side, but a trans is across. Like trans across, that's Latin. Okay, now we're moving on to carbohydrates. And carbohydrates are a fancy word for sugar. Sugars, which are carbohydrates, I'm just going to erase this. No one needs that crap. Sugars. They are hydrophilic, which means they are water-loving, which is not important at all. And you have...
It's 2 o'clock.
Just in case you were wondering. You have three basic types. You have your you have your monosaccharide, you have your disaccharide, and you have your polysaccharide. And what that is referring to is how many rings of sugar you have. So if we go back to the trusty internet and we look up monosaccharide, we search images, we can see some monosaccharides, and you can see how they each have one chain, and those are just three types. The main ones that I think you should know are fructose, glucose, galactose, deoxyribose, and ribose. Deoxyribose is in DNA and ribose is in RNA. Let's see if we can get a picture of those. Ribose. And the main difference is between the two is the amount of oxygen. Deoxy, meaning less than 1. You can see deoxyribose has hydrogen here rather than OH, because if it was ribose, it would have an O in addition to that H right there, but deoxyribose doesn't. And remember that we're talking about sugars here, so they are forms of energy. They store energy for bodies, and we use glucose in making ATP, and ATP is your energy, the molecule of energy that all organelles in a cell use to do any function at all. And so we have our monosaccharides, our disaccharides, and our polysaccharides. As I said, your monosaccharides are your galactose, deoxyribose, ribose, glucose, and fructose. Now, I don't know if your teacher is going to have you know them, but those are what they are. And what we're looking on the screen is what's known as an isomer. You have glucose and fructose, which have the same number of everything, C6H12O6. So 6H, 6 C's, 12 H's, and 6 O's. But they're isomers because they have different arrangements. They're the same formula, they're the different arrangements though. And I don't know that you're going to have to know that either. But as you can see, those are the different types, and they each have a different name. One is known, one sec. So what I'm looking up here is there are two things called an aldehyde and a ketone, and that just says the structure of that just talks about the structure of the sugar, and I'm not sure which one's which, so that's what I'm trying to find right now. Okay, I'm going to go out on a limb here and say that it doesn't matter. But I think fructose is your ketone and glucose is your aldehyde. And the way you know the difference is you have the double bond O sticking up there versus sticking out there. So ketone, aldehyde. Alrighty. And those are your carbohydrates, which are sugar, which is very important for energy. And then if you want to know some other ones, the disaccharides, you have sucrose, maltose, and lactose, and those are actually made up of monosaccharides. If you care about polysaccharides, those are your starch, cellulose, and glycosin. And those three are for structure. Cellulose, you've heard of. Starch is very important in structure of potatoes. And then glycogen, is used for energy storage in plants, I think, in animals. In It's in animals, so we use it. Glycogen. Alright, we're done with that. And then next we have hydrocarbons. And hydrocarbons are, I don't know what you need to know about them, but they are basically they're the simplest organic compounds. And organic compounds is a fancy way of saying molecules with carbon in them. So they're the basic, building block of life. And this is a Lewis structure, Lewis structure, for methane, which is the simplest one, which is just carbon with 4 hydrogen on them, and that's written as C4 sub H, which is CH, and then well, you get the picture. It's sub 4. Yeah. And then 4. That's basically what that is. And there's a bunch of different types. They can be arranged in rings. They can be branched or unbranched. You can have double bonds, single bonds, all sorts of things. But hydrocarbons are fuels. Hydrocarbons are 2 times as energy rich as carbohydrates. And they are used in fuel for cars. There are hydrocarbons in gasoline and octane. There's also hydrocarbons in lipids. As I said earlier, of course there's nothing there. The lipids here, these are hydrocarbons. As you can see, it's a bunch of carbon and hydrogen. And that's basically what an organic compound is. That's basically what a hydrocarbon is. And that's basically all I've got for you today. Yeah.